So I'm Christina Faust. I did a master's and bachelor's here at the University of Georgia in ecology, and I was the second graduating class from the Odum School of Ecology, and I'm very grateful for that. But many do not know that I actually am from Athens, and I first started at the Institute of Ecology with Allison Roy, who's in the audience, and I got a summer internship, and she gratefully took me on, and we did stream ecology in North Georgia. So we set up Tupperwares to collect insects that were falling into stream systems to look at how riparian zones affected drift feeding fish. And I also found some original notes that she gave me on my science fair projects. So this institute has been instrumental in my research career from even before I was at university. And from a very young age, I wanted to be a veterinarian, but Durso and Misha convinced me to switch over to ecology. And I did my most cited paper, still to this day, is based off of my master's dissertation, which I did up the road at Squidus. And much of what I love about the University of Georgia is the ability to work across departments. So Sonia was my major advisor, but she was so supportive of doing work on clams, filtering viruses out of the water, so looking at ecosystem effects on effects of transmission in avian influenza. And Ron Carroll has always been there and has always made fun of me for my diverse interest, but I think I learned it from the best. So when I got invited to this talk, I actually was out in Uganda, so I work in Lake Victoria now, but these hip waders that I wear now to collect snails were funded by a grant from the Odum School while I was here. They're still being used 12 years later, and they still don't have any holes in them, I'm pretty sure. So when I was here at the university, I took a diverse amount of classes. Pejro Hani was here. I still use his infectious disease book as my Bible, but I was also influenced a lot by ecosystem services. So I unfortunately did not overlap with Eugene Odum. He died the year before I started, but his legacy is carried on, and the faculty here is so diverse and wonderful that everyone has been great. So I am very interested in landscape changes and how these influence infectious disease dynamics, and I was very interested when I left here looking at population health, and I did some immunology work, but realized ecology is the place to do this work. So this is some work that I've done with Nicole Godinker, who's in the vet school, and we're thinking about this in a very simplistic way. So I use a variety of modeling and computational approaches, but always try to test these in the field, which I learned here. So if we think about pathogens transmitted between two separate hosts, in the most simple case, we can have two species, one that lives in a disturbed or human-dominated landscape, and another that lives in a core or intact habitat. So we can have many different pathogens that follow this trade-off, that there's two different habitat types, and spread either happens in a different habitat type or across a habitat boundary. But if we think about habitats converting in the most simplistic sense, as habitat is converted, we have an increase in habitat edge. And if we just use that habitat edge as a proxy for between species contact rates, we can create these really simple models that do a really nice job of predicting a lot of systems and exploring what host characteristics are most important in the emergence of infectious diseases. So so this habitat is changing from a core to a matrix, this is core habitat and this is matrix habitat in the most simplistic sense. And so we're converting it to a human-dominated landscape, and where do we expect spillover to happen the most? So this is the proportion of habitat converted, and this is just a stochastic simulation based off of an Ebola epidemic. And we see that at these intermediate levels of habitat conversion, we expect that the probability of outbreak is highest. And then we get a slight skew in the size of the outbreak, and that's dependent on various host factors. I'd be happy to talk to you about that. But this kind of fits a lot of systems that we look at. So in these intermediate disturbance places, we see both a spatial and a temporal increase in risk that then dies down and goes to an alternative stable state. But these are areas that we're really interested in undergoing land use change. And so some of my dissertation focused on long-tailed macaques, and they carry lots and lots of malaria species. But Plasmodium knowlesi in particular is emerging in human populations in Southeast Asia. And so some colleagues at Glasgow have looked at what affects risk of humans. So if you look at a human house, and then if you look at whether or not they're infected, so this is a case control study, and they've shown really nicely that within a 2-kilometer radius, you have to have 65% forest in that area, but you also have had to have had 14% deforestation in the last 5 years. So this is, you have to have enough wild habitats to support these reservoir species that are really good at transmitting, but you also have to be within flight distance of a disturbance so that the mosquito can then bite you with an infection. So this is a really nice way in that land use change is affecting infectious disease on a local scale, and it predicts really nicely emergence in some areas, but it doesn't predict emergence across the region. So, this figure is normally much better. So long-tailed macaques are spread throughout Southeast Asia. They have knowlesi throughout this range, but in dark shading is where you get really high rates of spillover of human malaria. So some of this is detection probability, but a lot of it is some other ecological thing that's going on. So we looked at many factors: rain, species composition, biodiversity, and everything. Nothing predicted it very well that we classically think of. So then I switched to looking at genetics of these reservoir hosts, and we focused on hemoglobin. So hemoglobin in humans is well known. Mutations of hemoglobin in humans is well known to be quite protective. So sickle cell anemia is the classic example. But if you go heterozygous for that, or homozygous recessive, then you're not in a good shape. But the macaques have co-evolved with malaria for 5 million years, and they have a much better solution. So we found that they have a huge amount of variation in their alpha-globin genes. So alpha-globin is a duplicated loci, and I'm happy to talk to people about it, but it actually duplicates further to about 27 copies in areas where you have really high rates of malaria transmission. And so they can fix their entire populations to have AQ, and AQ is a phenotype, but it's completely protective against malaria. So if you infect macaques from Bali, which is here, with malaria, they die within a week. And if you infect macaques in the Philippines with malaria, they survive just fine. And this is all due to this variation in hemoglobin that we think. So that kind of predicts our large-scale patterns that we're seeing with emergence. So the macaques that are in these areas that have co-evolved with malaria are able to have a much higher level of Plasmodium knowlesi, which then, when you have land use change, leads to spillover. But we get land use change in all of these areas of Southeast Asia, but we only see spillover in places where these macaques have adapted to coevolution with malaria. So and I'm happy to talk to people more about it later. So, like Alan said, I now work on schistosomiasis. So it's a great system, and I love it, and it's wildly complex. So we've got mammalian hosts that host the adult worms and go through sexual reproduction. And then the transmission is facilitated by poor sanitation, basically. So I collect human stool and hatch it into Myristidia and do genetics on that. And we also do a lot of snail surveys and collect Cercaria. But there are about 1 in 2,000 are infected with Cercaria, so it's a very laborious process. So the area where I work is in Uganda, and Uganda was the first country to start introducing control of praziquantel. So this is a drug treatment that's used to treat schistosomiasis. And we've been treating they have been treating for 14 years, but in my district, we've seen an increase in prevalence and also an increase in infection intensity. So this means the average eggs per gram that are being output in human stool is actually increased since baseline, which is quite alarming. So the kind of dogma is that we're having selection for resistance. But so this is a data set from 3 years that was collected by my PI, postdoc advisor. And if we look at treatment So we get before and 1 week post-treatment. The genetics of the parasites are very similar, which is expected. So a week after you're treated, the adult worms are dead, but you're still excreting parasites that look very similar to the pre-treatment. But at 4 weeks post-treatment, a lot of people are cleared. About 60% are completely clear, so you get no eggs, so you get no genetic diversity. But of the remaining genetics, you see a decline, and if you control for age and sex, it's a significant decline in genetic diversity. So it's having some effect in the short term on individuals, even when they're not clearing. But unfortunately, we get a complete genetic diversity recovery. So the amount of diversity that you have inside of you completely recovers, and the intensity of infection recovers within 6 months. And we're actually now, we've got 4-month data coming in that says it's recovering within 2 months. So it's rapid reinfection in these places. And what we think is going on, but it's still an ongoing work. So this is part of Lake Victoria. So Kenya's over here, and we work in the eastern region. And these are some of the villages where we've worked, and we get complete gene flow between these regions. So we think what's happening is you're treating a village, and then you're moving along and you're treating another village 4 weeks later. But in that period of time, humans have moved back and forth and kind of reseeded the genetic diversity of these. So these are about 100 kilometers away, but we see no genetic structuring in parasite populations. So we have high rates of human movement that's facilitating gene flow. We also have, we think, a high refugia of systematically untreated people, which is quite unfortunate. So this is age across the x-axis that goes up to 60 years old. And in dark red are people that have never been treated. So these people have had 14 chances to be treated, and they've never been treated in their entire life. And the dark blue were treated in the last year. So we're getting really poor coverage, and also systematically untreated people that have huge amounts of genetic diversity. But kind of what I'm interested in, what we're doing this summer, is looking for selection for hybrid schistosomes. So this drug treatment should have an effect on the population, and we're seeing no effect. And we're using very basic markers, microsatellites. So now we're going in and we're doing whole genome sequencing to look for evidence of hybrid schistosomes in these areas. So this population in this district, so kind of this, I'm gonna mess this up. So this whole district where we have has increased tenfold in the last decade, and that's when MDA has started. And it's gone from 80% deforestation to 80% forest to 5% forest in that area. So this is what the community looks like now. It's all smallholder farmers, tons of rats that can carry human schistosomes and also have their own. So we're looking for evidence of adaptation by incorporating other species genes in these populations. So everything I do is informed by ecology. I did my PhD elsewhere, but everything I needed to know I learned here. So you can tell Princeton that. And I think this department does an amazing job of predicting epidemics moving forward, and there's some really exciting work coming out. And I now am moving more into molecular ecology, but everything has been informed by my time here. And I want to thank Nicole Gunning here especially for some of the land use work that we're doing. And I learned how to collaborate while I was here, and everything I do is never at my own institution and not involves many other people. So we work with Warwick and Oxford for the globin gene stuff, and then now I'm working with the Ministry of Health and my PI, Poppy Lamberton, for the schisto. And yeah. Any questions?